All right, when we last met, we talked a little bit about Gregor Mendel's experiments, his basic setup. He is pollinating plants by hand, crossing white and purple flowers to see what happens, okay? Now, the trick is to look at the patterns. He always sees a 3-to-1 relationship when he does it in the way we described, and he now has to figure out what would account for that. What would have to happen to account for that? And he comes up with this idea that there's these particles, what we might now call genes, right, that are passed on, and they must come in pairs, you know, like we see here, big P, and big P, little p. They come in pairs, but when gametes are made, the alleles are going to segregate. We know that word segregate is a word that has pretty negative connotations in our history, but segregate means to separate. So what he means by that is, if in this adult there's a big P and a big P, in the gamete there's only one of them, right? They separate. There's only one in a gamete. And therefore, if there's only one from this parent and then only one from that other parent, they're going to come together, and he can say, if that would be the case, we'll get a 3-to-1 relationship. He did it with probability, just doing basic probability, but you can visually see why you would get a 3-to-1 relationship doing a Punnett square. And I bet you all know how to do these, but let's do it here, crudely here. So we put the gametes from the purple parent, we'll put one here. Big P, oh, sorry, hold on, this is a bit difficult. Big P, oh, come on, and big P and little p here. And then we'll put big P and little p here. So if this gamete and this gamete cross, we get big P, right? If this one and this one cross, we get big P and little p. If this one and this one cross, we get big P, little p. And if this one and this one cross, we get little p, little p. So that's not telling us what would happen if we made 4 babies. It's giving us probability here. If we have 100 babies, how many purple will there be? How many white will there be? We can kind of figure that out. And what we can see here is that this is purple, this is purple, and this is purple. So that's 3 purple for every 1 white offspring produced, is what he would predict would happen if this is how genes are passed on in this segregation model, where there's 2 copies in the adult, but when they make a gamete, only 1 copy goes in. One is dominant, one is recessive. Then he can predict this pattern. The same thing can be done just using math, the idea of probability. And if you've taken any stats, I'm sure you're familiar. But let's say you have an organism, once again, that's, in this case, we have big R, little r, right? And so a sperm or egg will have either a big R or a little r in it because of segregation, because of meiosis, basically. He didn't know what meiosis was, right? But because of meiosis. And then, of course, we have fertilization. Who's going to mix with who, shown in the Punnett square. So the chances of getting a big R or a little r in a gamete is 50-50, just like a toss of a coin, heads or tails, 50-50. So here, 50% heads, 50% tails, 50% heads, 50% tails. And then the question is, what are the odds that that heads, if you flip 2 coins at once, that one will be heads and the other will be heads, right? That's what it's asking. What's the chances of that? And to do that, you use multiplication. You take 1/2, or 0.5 times 0.5. So 1/2 is 0.5 in decimal form, right? That I think better in decimal than I do in fraction, but 0.5, 0.5. So the chances of getting 2 of these at once, you figure that out by taking 0.5 times 0.5. And that's 25% of the time. What's the chances of getting this and this combination in this case? 25% of the time. Here again, 25% of the time. And the chances of getting 2 tails at once, 25% of the time. So notice that you're going to get at least one heads 3 times out of 4 still. So a 3-to-1 ratio. So everything Mendel is doing is probability. What are the odds of having this and this happen at the same time? And that's what a Punnett square lets us do visually. Now, if we look at what we know now and update what Mendel is studying, we know that the flowers contain chromosomes. And at a particular locus, at a particular location on that chromosome is a gene for flower color. And that gene can come in different alleles. So in this case, we have the purple allele, and in this case, we have the white allele. So Mendel didn't know that, but he figured out that there were some kind of particles. That's as far as he could figure it out. And one was dominant, one was recessive. We can go even further and zoom in on that gene, and what we now know is that in Mendel's flowers, for purple flowers, here are the base pairs involved in that gene, or at least part of that gene, right? The DNA. And notice that in this part of the gene, we see it starts with CTA. In the white alleles, there's a mutation, and we have ATA. There's a single base pair substitution there. And what's the consequences of that? Well, when this gene is expressed, when it goes from DNA to RNA to protein, there's a protein right here. That protein is part of the pathway that makes purple pigment. Okay? But when there's a mutation here, that enzyme's the wrong shape. Structure and function are really important. And when that enzyme's the wrong shape, no purple enzyme is made. So instead, you get white in that case. But as long as you have one copy of the purple gene, you can still make purple pigment, and the flower is purple. So what does he figure out here? Each parent contributes one allele per character. Each diploid organism has two allele per characters. We've said that over and over again, right? Each one has, let's just say, big P and big P. They each contribute one, though, so when they make gametes, only one of those ends up in a sperm or an egg. Some traits are dominant, some are recessive. We've already established that. And so what he does is he looks at these patterns and he notices this 3-to-1 relationship in that F2 generation over and over again when he does it in the way we've described. And he says, okay, there must be some kind of separation where each parent has two of these traits, but they only pass on one copy of them. He calls that then the law of segregation. Very important Mendelian concept. We can now describe it and explain it because gametes are haploid. They have one copy of everything. Individuals are diploid, but they make gametes through meiosis. So what we can say is alleles separate when gametes form, and each gamete only contains one copy of each allele. They are haploid. Mendel didn't know that. We're adding in what we know now on top of Mendel's framework here, but that's what's happening. Alleles separate, meaning if your adult is big P, little p, the sperm you make might only have the little p in it. It's not going to have both. If it has both big P and little p in it, then you've had a non-disjunction event, and you might actually have an issue like Down syndrome or something like that occur. All right, so the last slide on this video segment, we'll just look at the terminology then that goes along with Mendelian genetics. So we have alleles that so the letters are alleles, right? Alleles are versions of the gene, and we can be purple or white in this case. If it's capital, it's dominant. If it's lowercase, it's recessive. And then these terms here have to do with genotypes. So these words up here have to do with alleles. These have to do with genotypes. If you have two copies of the dominant allele, you are said to be homozygous dominant. All right, let's break that down. Homo means same, and zygous means the zygote. When the zygote was made, it's diploid, right? So homozygous means you have two copies of the dominant allele. If you have a big P and a little p, a copy of the dominant and a copy of the recessive, you are hetero. Hetero means different. Heterozygous, so that means you're not true breeding, right? You're big P, little p. If you have two copies of the recessive allele, you are homozygous recessive. That is your genotype. Those are very important terms to know and learn well, because you'll hear about them probably in every course throughout your biological career. All right, we'll move on with the next video where we'll continue this process.